All right, I will.
I would have brought the kids because they're all doing something. Mike's driving them around.
Good evening. I want to welcome you all to the Frontiers of Science. My name is Frank Brown. You probably all have seen me before here. I'm the Dean of the College of Mines and Earth Sciences that jointly sponsors this series, the College of Science. In spite of our best effort to get the word out at the last minute, some of you may still be expecting to hear from Dr. Jess Adkins tonight, but due to circumstances beyond his control, he had to cancel, namely because he has a problem with his back and he can't travel at all. I think he's flat. I can appreciate that very well, having laid flat. We are extremely lucky, though, to have been able to welcome Dr. Thure Cerling to speak in Jess Adkins' place. Dr. Cerling is a member of the University of Utah faculty, both in biology and in geology and geophysics. He arrived in Utah in 1979 after undergraduate work at Iowa State University and then graduate work at the University of California, Berkeley. He's a distinguished professor in both of the departments, and he's also a member of the U.S. National Academy of Sciences and recipient of the Utah Governor's Medal for Science and Technology. And he has served for many years on the U.S. Nuclear Waste Technical Review Board to very good avail. Thure and I met in the 1970s, and we've enjoyed a series of wonderful collaborations in the Turkana Basin in northern Kenya and in southern Ethiopia. I, in fact, got back from there yesterday at 4:30 or so after a 27-hour flight from Nairobi. Thure and I began working on volcanic ash layers there when there was difficulty correlating between different parts of the basin. But his love has always been in isotopic studies, and I can think of no one who has such an incisive intellect when it comes to problems. He often comes up with first-order solutions on the spot, and then he drills down to show that ever more information can be reliably extracted from his data. I have no idea where he will take us tonight, but even in my jet-lagged state, I will doubtless learn something new. So please welcome Dr. Thure Cerling.
Yeah, thank you, and I was too looking forward to hearing Jess Adkins speak, but we'll have to wait for another time. And so Jess actually called me on Friday because he couldn't get a hold of Frank because Frank was in East Africa and said that his back was out, and like Frank, I also had a back out, and so I really feel it's definitely those of us who have bad backs, and we need to step up and take care of our fellow bad backers. So I'm going to talk tonight about environments of human evolution, and Frank mentioned that he and I first met almost 40 years ago. It was in 1975 in Salt Lake. I was working actually for the Anaconda Company looking for uranium, and I called Frank, who was on the faculty here, and we started some discussions and have had a lot of coffee and discussions ever since. And so in a way, this is kind of a theme that's been running through my entire career, learning about human evolution. And I guess for me, it actually goes back a long time. I grew up in the Midwest and was intrigued by the problem of human evolution, in part with the conflict, or apparent conflict with religion. And so I remember as an undergraduate having the opportunity to go to East Africa and to the site where we'll be talking about today. And that opportunity came about, and if there's any young people here, you should, listen to these words. What I found out then is when somebody says, do you want to go somewhere, you raise your hand and say, yes, it'll probably be a worthwhile journey. So I was an undergraduate working in a lab doing drafting and Carl Vondra at Iowa State University said, oh, so-and-so can't go to do mapping in East Africa, and he was looking for a particular graduate student who says, Alan, I need somebody to go to East Africa. And this guy said, no, Dr. Vondra, I want a paying summer job. I want to buy a car. And so I said, I'll go. And for no money, I went, and I've been going ever since. So could we dim the lights a little bit? Is that possible? We'll see if it is. So I'm going to talk about isotopes, and one of the things I've learned in my long time is that there's often a misconception. Well, I'll start with somewhere first, I guess. So in science, we often think that one of the things we like to do is get the cover picture of journals. And this is an article that I was associated with a couple of years ago, and it was about these man-eating lions of Tsavo, and this was a big movie a couple of years ago. And so we wrote this paper. We got the cover of the Proceedings of the National Academy of Science, and this had been made into the first 3D movie ever. And so this is, in some ways, possibly as high as you can get in science. You can make the cover of Nature or Science or the Proceedings. But then, about a month later, I found out that we really hit the peak of things. And we inspired a New Yorker cartoon with our paper. So that's as good as it gets, if you can inspire a New Yorker cartoon. So I'm going to talk about isotopes, and I've discovered over the years that some people have forgotten what they learned in kindergarten about when they took chemistry. I know you all took chemistry in kindergarten. And so isotopes are just different ways of thinking about atoms, and we remember that the nucleus of atoms has neutrons and protons, and we all remember the periodic table of the elements. I mean, I'm sure you all know this by heart, at least the image, but what I like to think of isotopes as it's a third dimension of this periodic table. So you're generally familiar with the periodic table of elements. There's some sort of order with electrons and things and it is based on the electrons, that the chemistry of the different elements. And so the isotopes are sort of an individual species, as it were, of the different atoms. So for carbon, we have 3 isotopes that naturally occur on planet Earth. Carbon-12. So because it has 6 protons, it's carbon, but it can have several different neutrons. The neutron has mass, but no charge. And so it can have 6, 7, or 8. And so we've heard of carbon-14. That's the radioactive isotope of carbon. But there's also carbon-13, which is a stable isotope. And so today I'm going to be talking about stable isotopes. And so we're really interested in the very tiny variation of carbon-13 relative to carbon-12 on planet Earth, or oxygen-18 to oxygen-16, or nitrogen-15 to 14, hydrogen. So you can see hydrogen is 99.98% of the normal hydrogen, but there's this teensy little bit of hydrogen with an extra neutron, and so it's that difference that we want to utilize. And that difference in mass, anybody who's moved a friend to an apartment on a second or third floor kind of realizes, you leave all the heavy boxes for the other people and you take the light boxes, and you say, I moved 16 boxes and you only moved 14. Well, you had him move all the heavy ones. So there's a separation that occurs. Even though the chemistry is the same, there's a separation that naturally occurs, and that's what we want to use today. So these are not radioactive isotopes, but they're isotopes nonetheless. And this is so carbon. So for carbon, on planet Earth, the natural variation of carbon-13 is from 1.04% to 1.14. So our entire space that we're examining is in that that 0.1% variation in carbon-13. And so we use this terminology, which we call the delta notation, for carbon-13, and we just simply look at the ratio of carbon-13 to 12 in something versus some standard, and for historical reasons, we just kind of compare it to 1. So we're looking at two ratios that are almost identical. So a negative value means that it just has less carbon-13 in it than something else. And if it's really negative, it has even less. So that's all you really need to know. And this is true for all of the different isotopes we talk about. So negative values means there's just less and less. And positive means there's more and more of this, of the rare isotope. So that's what you need to know on the chemistry. So we're going to explore today this evolution of humans from sometime after we split off from the chimpanzees, which is probably about 6 million years ago, and in East Africa. So when I was an undergraduate in the late 1960s, there were enough decent specimens that every single one had a name. There was Rhodesian man and the tongue skull and so on. And since I began working there, the first time I went was in 1971, we found literally many, many hundreds of fossil early humans in East Africa. So now we have this branch, and we're not going to worry too much about the details, only a little bit as we get further about distinguishing differences between some of these organisms. But I'm going to explore today the job as the geologist is the question always is, well, what is the environment really like that these creatures were living in? Was it like on the right, on the far right, was it an open grassland? Was it a little bit more closed? As we move to the left, we get more and more closed. Were they living in something like we find on the left today, which is what you might find a chimpanzee living in, or something way on the right, or something in between? So those are some of the questions. And there's a lot of people that I have to thank, and there's many students from here at University of Utah. There's colleagues from the National Museums of Kenya and from Kenya Wildlife Survey and so on. And so I just want to recall that there are many colleagues and friends who have helped me on this. And then at the bottom, there's a number of organizations that have facilitated the work in many ways, sometimes by actually providing money, and others by, for instance, the Stable Isotope Ratio Facility for Environmental Research is the lab at University of Utah where I collaborate with Jim Elringer. So these are some of the cast of characters, and all of these specimens save one. So the one on the very lower right was discovered in 1969 at Lake Turkana, and then all the other ones have been discovered since 1969 in Lake Turkana. And this is kind of our cast of characters. We've got many, many more fossils. Tens of thousands of fossils have been found, including, of course, many different things. So the hominid fossils, the early humans, are the rare ones. This is a bit of the geology. It looks a lot like some of the badlands in the western USA. There's a bunch of geologists walking on the bottom there. One of those is Frank Brown, the tall one, I imagine. And in all of these pictures, there's a layer of volcanic ash. And Frank has spent many years trying to sort out these volcanic ashes, and those provide unique time horizons. So this basin is extraordinary because it has such exquisite dating control. Here's some of the cast of characters. Ian McDougall, who's been involved in the dating. Meave Leakey on the upper right, who's a paleontologist. Richard Leakey, who was former head of the museum. Berchet Haileb, former student here. Ben Passy, student here, so on. And Frank and I in an anthill for scale. So you think Frank is tall, think of these anthills. So here's the basin that we're working with. There's a large lake in the middle of it, Lake Turkana. It's about 250 kilometers long, so it's And you kind of have to drive around the lake if you want to get from one side to the other. And people often don't have a sense of scale. So here's scale, equivalent distances. Moab would be down there at the bottom, Salt Lake a little bit above the middle, and Pocatello. And I've also shown the location of all the gas stations in the region. So all the gas stations are in that red dot. And so there's some, let's just say, some logistical challenges. It's also in 3 different countries. So besides a river that has only one bridge in its 400 or 500 mile journey and a large lake. No, there's one bridge further up. Remember, that's the one where we were told we had better put our cameras away. Anyway, so there's some logistical challenges, and that just makes it interesting. So this is what Frank alluded to. There's these layers of volcanic ash, and this is what Frank and I got started on. Again, when Frank and I began working on this project, to actually date these layers of volcanic ash, we needed to nearly fill that bottle full of those little tiny crystals on the bottom slide there. But now we can date just a single, basically one of those single crystals about the size of a sand grain to actually get a date. So that's made things nice. And Frank has devoted much of his career to sorting out the geology and allowing the story that I'm going to tell by providing the dating. And there's many methods for dating. We have potassium-argon dating. We have magnetic stratigraphy dating. The Earth's magnetic field flips back and forth. Frank has correlated some of these ashes out into the deep sea, where they can be dated with other means. And so we've got many, many dates and many of these volcanic ash layers, so that this is truly the best place to study human evolution, because we have so many dates and we can kind of understand the progression of things. And so those are some of the stratigraphic areas, and each of those has to be treated really separately in a separate expedition, just because of these logistical details. So these are the questions that, as an isotope person, I get asked. What was the vegetation like? How hot was it? How much, what did animals eat? How dry was it? And then I was even asked once, and I'll come back to this later in the talk, a colleague of mine at Olduvai Gorge, showed me a hand axe, and he said, well, was the hominid who made this sitting in the shade when he made this? And I said, oh my God, give me a break. And so I said somewhat flippantly, if you tell me the time of day, I can tell you if he was sitting in the shade. And we'll return to that question. So one of the big players is carbon isotopes for me. And I was lucky here at University of Utah to have Jim Ellringer as a colleague. He's sitting over there on the far side. And the story has to do with 2 different forms of photosynthesis, which we simply call C3 and C4, and that's because the first carbon atom, that as it's fixed, forms a molecule that has either 3 carbon atoms in it or 4 carbon atoms, and so we just use that terminology. The important thing for this story is that the trees and shrubs and most herbs like, say, rosemary and thyme and tarragon use the C3 pathway, and the tropical grasses use the C4 pathway, and they're very different in their isotopes. And we can see that easily different. So we want to use that difference to tell us different things about the different environments. So there's almost a pure C4 environment on the right, with those 2 elephants standing around in the tall grass, and an almost pure C3 forest on the left. So this is a dry tropical forest. And where, in terms of going to the grocery store or something, the C3 plants, that's the early photosynthetic pathway on Earth. It's been around for 3 billion years. Almost all dicots, those are the plants that when you plant them and 2 leaves come up, like when you do the planting of the bean experiment in 4th grade. It's also the cool season grasses. And so when you go to the grocery store, it's the vegetable, in the vegetable category, the beans and the rutabagas and the carrots and the peas are all C3 plants. And if you go to the meat counter, well, then it gets complicated because the meat depends on what the animal ate. So that's important for our story later when we think about early human diets. And C4 plants are the tropical grasses and sedges, and it just has to be warm during the growing season. So each of you do a little experiment in your own yard every year with C3 and C4 grass competition. And in May, when things are nice and green and fresh, in my grass, at least the Kentucky bluegrass does really well. And it kind of is really tired of life about 4th of July. But another grass does really well, apparently, in spite of the fact that it's not getting enough water, and that's crabgrass. Crabgrass is a C4 grass, and it does quite well in spite of the more problematic conditions. So in the grocery store, the corn is a C4 plant, maize, sorghum, sugarcane, and then those other things, millet, teff, and fonio are some African grasses. And of course, in North America, there's lots of native C4 grasses, and they're most abundant in Texas, and they pretty much are gone by the time you get up to North Dakota. So shade, how can we deal with this problem of shade? Because this is the question that, it is important if you're thinking about trying to evaluate what humans, how they might behave on the landscape. I know when I'm trying to do field work in East Africa, shade is really, really important. and my day has a lot to do with where the shade is. So here's 3 examples. Something that I've quantified as 8% shade by looking at satellites, measuring things on the ground, about 20% shade, and except for where the truck is, 99% shade. So we'd like to use this ecological structure. And we might think, well, shade really is, it's actually woody cover is what it is. It's something that's tall enough to help out. So that could be an indicator of the ecosystem structure. And so the way that we approached that was to say, the woody structure are the trees and the shrubs. So we can call those shade, and then we have everything else is not shade. So we'll just try to plot shade versus not shade and see if the carbon isotopes can help us. And so we go to the field and we look at about 75 or so sites in the tropics, and this is Africa, Australia, and so on. And we measure, and in the end it's actually over 5,000 soils that we've collected, where we've collected things from below the canopy and in the open and try to quantify this, and we find that, it actually doesn't, the plants don't really care that much over, or the soil doesn't actually care that much about whether it's growing underneath the tree or not. If we're underneath an open canopy, that turns out grasses can grow underneath there, and if we're in a really closed forest, it turns out that in the gaps there's so much shade anyway that the plants aren't really quite that aware of it. And so it turns out that the measure of the carbon isotopes in soils tells us about shade. So we, with the help of Kenya Wildlife Service, in here, and then my colleague Michael Bird doing other tropical sites, we look at a whole variety of sites that we might think of as analogs, and we quantify the woody cover based on satellite imagery, walking around taking pictures, walking around taking notes in the field, and come up with a relationship that shows the carbon isotopes in soils versus the fraction of woody cover. So this is kind of a ground truthing validation, and then we can calculate at any carbon-13s, and sort of in this schematic diagram, if we have a carbon-13 value, we can calculate a kind of a Gaussian probability function for the shade that would describes this value. And so we can do that in modern soils, and it turns out that in East Africa we have a lot of fossil soils. This is fossil soil from the Turkana Basin, and so this is a big layer of volcanic ash. So we know exactly when this soil was buried by the volcanic ash. We know its age. We can find organic carbon and calcium carbonate nodules that formed in the soil, and from those we can determine the carbon-13 of the soils. And so I just show some trends with time. So this is 4 million years ago, 3 million, 2 million, 1 million years ago in the Turkana Basin. And this is, the green here is in the Shungura area, which we think is sort of an axial river system. So these are the soils forming right near the river. And farther away from the river is this reddish stuff. It's actually more open, which is what we find today. Near the river, we have these more closed habitats. And then at about 1.8 or so million years ago, they kind of come together. And so we can actually then use this to quantify, well, what was this ecosystem like? This is 50% woody cover, and this is 20% woody cover. So we're between 20 and probably 60% woody cover, and this stuff is quite a bit more open. And that has consequences if you're an organism out on the landscape. Certainly if you're trying to get away, for instance, of something that might want to eat you, or if you're trying to catch something that you may want to eat. And these are some of the famous fossils from the Turkana Basin, and where the ecosystem that would most likely closely describe them. And then I've just here tried to put some photographs. This is 20% open cover, 40, 65, 80%. We never find anything like this. We rarely find this. This is the most common sort of habitat. Okay, and this actually is about less than a football field length away from a really major river in East Africa. So, we probably have this sort of habitats right near major rivers like the large Omo River, which flowed through the Turkana Basin for many millions of years. And likewise, this other site over here, this is also about maybe a kilometer away from a major river in Kenya. So as you get near the river, it would tend to be more like this, but it's a very narrow zone that we don't find too much evidence of. But we do occasionally find it down here. And we can do this for all the fossil sites in Africa. And when we do that, this is the Ethiopia region where Lucy came from. And this is the Omo-Turkana Basin. And these two basins together provide probably about 90% of the fossils, the early human fossils, that we can actually date and say how old they are. So this is the real place to learn about human evolution. And what we see is some trends. And we see, but basically, mostly woodland, bushland to a grassland. Never, or very rarely, do we actually find these open grasslands. Those are really pretty uncommon. Likewise, in the Turkana Basin, in this lighter yellow-blue, or lighter blue, is Shungura, and this is the Nachikui cuvifora. So this is near the river, and this is away from the river. So just to, again, give you a visual sense. So altogether in East Africa, we've analyzed close to 1,400 paleosols from many sites. And we can say, well, what is, maybe 20 or so % is this really open grassland. And these are almost all the youngest soils in the 4 million year record that we have. So there's not much of this, maybe 20%. Most of what we find is characterized by something more like this, okay, where it's kind of a savanna, but a pretty much a wooded savanna, environment, and not much in these woodland environments. 15% of all of these things we've looked at, and nothing at all in this true forest environment. We really, in this heavy, heavy, dense bush, we just don't see much evidence for that. And as an example, here's just 3 sites from Africa. A site called Kangera that we think would look like this. A site called Aramis that would look something like this. And this is one of the most densely wooded sites we have an example of in East Africa is something much, much more like this. Okay, and again, in each of these sites, we've measured at this site. This open area in the foreground is 5%. In the background here is a very narrow, about half a football field wide, riparian, so tree corridor right along the river. So we think these were present, but as very narrow corridors in many of these sites. And this is what they look like from the air. And again, you can imagine that if you're a hominid and you want to get from here to here, from here to here, here to here, this is what you have to travel around to go any particular distance. So you get more and more wooded. You have to, you certainly, even in this sort of a habitat environment, you can't really stay in the trees all the time. You have to get down on the ground and get in between if you want to, even if you want to get all of your resources from a tree. Likewise here, you've got to spend more and more time on the ground. So we can actually begin to say something about the ecosystem structure in a way that sort of makes useful analog sense. Another question always is how hot, and this has been a problem that as geologists have struggled with all the time. We always like to say, and when we're reconstructing an environment, well, it was hotter or it was colder, but we'd really like to know, well, how hot? Exactly, what sort of temperature regime can we do? So here's 2 environments in Kenya. This is Lake Nakuru in the Rift Valley. The mean annual temperature is about 17 degrees Celsius. And here's Lake Turkana. This is what it looks like where the mean annual temperature today is 29 degrees Celsius. This is hot, hot, hot. So this gets us to this question, I guess, about tools again. So where were these guys when they made these kind of tools? And were they sitting in the shade? Well, let's explore this hotness idea. This is the mean annual temperature for different land parcels on the globe. And it's actually done by just by latitude, longitude. Salt Lake City is here about 10 degrees Celsius. Los Angeles, nicer, about 16. Death Valley. Here's Lake Turkana. It's in this box right down here. So it's in the top tiny, whatever it is, one of the hottest mean annual temperatures on Earth. And so what we use, one of my students who got his PhD here went to work on a newly developed thermometer in geology, which we call the Delta 47 thermometer. And thermometry in geology goes back to just before I was born. There was a classical paper, actually it was a classical presentation done in Switzerland by a guy named Harold Urey. And Harold Urey was giving a talk in the either chemistry or physics department at ETH in Switzerland, and was a good example of why different departments should go to other people's seminars. Geologist was there, and he said, Dr. Urey, if what you're right, if you're right in what you're talking about, you've invented a thermometer for geologists. The thermometer is as follows. Again, either in kindergarten or first grade, you learned that all chemical reactions are functions of temperature. So sugar is more soluble at higher temperature than at colder temperature, and all chemical reactions have some temperature relationship. So the temperature relationship we're interested in is calcium carbonate, calcite, with one heavy isotope, reacts with water with a light isotope, and trades it for, so this heavy isotope of oxygen in the calcite gets traded with the water. And we can write a chemical reaction, which we learned maybe in third grade, I forget when. But because it's a balanced chemical reaction and it's at equilibrium, it has to be a function of temperature. So this is the basis of the beginning of geothermometry, and the problem with this is you need to know the ratio of the isotopes in calcite, and you need to know it in water, and we don't have fossil water. So the conundrum for geologists for decades and decades is, how do we make a good guess as to this number? What can we do to get around it? So about 5 or so years ago, a guy named John Eiler at Caltech said, oh, I've got a better idea. This is simple. Let's take calcite and react it with calcite to make calcite and calcite. Wow, that's really good. And he said, but there's a small detail, and the detail is we're going to look at this oxygen 18 and this carbon 13, and we're going to look at it when those two rare isotopes are coupled together. So we have 4 different molecules of calcite that have different weights. And this one has a total weight. The carbon dioxide that we get from this has a weight of 47, so that's why we called it the delta 47 thermometer. So it's a really simple chemical reaction. Anybody can remember that. But the measurement is really difficult. And so fortunately, one of my great graduate students did a postdoc. Ben Passy did a postdoc with John Eiler, and with another graduate student I had who also went to work with John Eiler. And Ben and Naomi got married, and now just have a month-old baby. So great things happen in graduate school, I guess. So they collaborated to work on this thermometer and apply it to the Turkana Basin. So we're interested in this rare isotope, and all we need to remember is this name, delta 47. This is the coupling of these 2 rare isotopes gives us absolute temperature without needing to worry about it. And it's a lot of work, and Ben is particularly persnickety about getting things right in the lab. And so we apply it to the Turkana Basin. This is a modern soil, and this is calcium carbonate that precipitates in the soil. I've got other students who are working down in Boulder Mountain on the black boulders and the carbonates there. We're doing similar kinds of work. So we apply this to the Turkana Basin, and what do we get? All the soils say they were hotter than today. So today is 29 degrees mean annual temperature, and we're getting temperatures up to 40 degrees Celsius. So that's a bit unhappy. That's the hottest place on Earth already, and it says it's even hotter than that. So Naomi and I buried a bunch of these little data loggers that you can put in your own yard. We buried them in soils, and then with the help of the Kenya Wildlife Service, we instrumented a whole bunch of national parks. So we did this in lots and lots of soils all over Kenya. What we find for Turkana Basin, for this one time interval, mean annual air temperature 31 degrees Celsius. The temperature is 36 degrees, so we're okay. So mean annual temperature in the soil is way hotter if you're in the open. So Illoret, mean annual temperature 29, open 36 degrees, in a really skimpy forest. It's only 30 degrees. It's basically the same as the mean annual air temperature, and we find this true everywhere. Nairobi, where it's much cooler, the open areas are still much hotter, and the forests are basically, that's where it's nice and cool, and we all know that from camping and hiking in the summer. Go to the shade for trees. So one of the students that I had on this, Will Mace, working with Dave Chapman, said, well, wow, we've got all of this data. So this is our soil temperature envelope for every day. And here's a typical day. So this is at 5 centimeters depth red, and then at 10 centimeters green, or 2015, and at 25 centimeters blue. And so Will Mace, working with Dave Chapman, said, oh, it's easy. We can input a temperature that uniquely satisfies all of these data, and we can do it for every day of the year. So this is what we do. So we calculate what the skin surface temperature of the Earth is every day for a whole year for 3 different sites, an open grassland, bushland, and one of these riparian forests. And here's what the forest looks like. The hottest it ever gets is low 30s Celsius. In the open areas, it gets into the mid 40s. We even get up over 60 degrees Celsius up here. This is 135 degrees, 140 degrees Fahrenheit. Up at Lake Turkana, we even get ground temperatures up to 160 degrees Fahrenheit. This is potentially lethal. And I have seen people foolish enough to try to walk down to the lake without shoes and have literally blistered their feet. It's hot. So Will said, well, let's call this we could call this interval, let's call this what's the average day look like if you're out here, in here, or in the forest? And this is the surface temperature. This is what your feet would experience if you're standing on the ground, like you're going to say, be a hominid making a tool. Here's the composite day for 3 months, June, July, August. So it's 6 o'clock in the morning before the sun comes up. It's coldest, as you might expect, and it gets really hot. And then in the afternoon, it gets cool again. And now it's night. And it gets cooler in all the 3 different places. In the forest, not very much. Not so much temperature change. So now we could address this question of where was the guy making the tools? Well, let's just say 40 degrees Celsius is an important time to think about it. That's 104 degrees Fahrenheit. That's too hot to do anything, even except go in a swimming pool. So 40 degrees Celsius, if you're in the open, the only time of the day, well, it's dark until 6 o'clock and the sun comes up, and by 9 in the morning, it's 40 degrees Celsius. And so you got to get back in the shade. And then you got about an hour before dark when it's cooling down. So if you can actually see and make the tool, if you can tell me what time of day it was, I can tell you if he's in the shade. Okay? And the other bottom line is that this has something, also an important issue about intelligence of hominids. If they're as smart as goats, they're in the shade. So, middle of the day, these guys are always in the shade. They know where to find it. Another question is how arid is it? How dry is the environment? And Naomi Levin was one of my students evaluating this problem. And the conceptual model is that we have what we call meteoric water, which is basically rainwater, river water, groundwater. It has not seen the atmosphere very much. In a dry environment, the light isotope boils off more easily, just like going up to bringing the heavy boxes up the stairs to get to the second floor apartment. So the heavy isotopes get left behind. So as we get a higher water deficit, so decreasing humidity, we have more and more evaporation. So how can we do this? We need something that records meteoric water and something that records evaporation. So our approach is we subtract an hippopotamus from a giraffe. This is a geologist's way of doing it. So our notion is we can look at water deficit, which is the amount of missing water, as it were. So how much evaporation there is, potential evaporation, compared to how much rainfall we get. And we go from a gradient from rainforest to Lake Turkana in East Africa, and we just look at it and think, okay, this represents our evaporated member, and this represents our river notion. And that difference, what we find in Turkana is everything is between these two kind of environments. I skipped over a slide there a second. Okay, so here's the Ituri forest is here, Nairobi is here, and what we find in the fossil record is everything looks pretty dry for the Turkana Basin. And this is true in other regions as well. So we've answered a bunch of stories so far. We've been able to say something about paleo shade. For vegetation, it's not these kind of things. It looks like these kind of environments. Okay, a lot of open environments. Perhaps if there's a narrow forest, it's in one of these narrow riparian corridors of a river flowing through deserts. And then the last thing I want to touch on is diets. We've done a lot of work of diets on animals, but I want to think about human diets. So think of your own diet. These are C3, healthy C3 foods, and perhaps not so healthy C3 foods, and C4 foods. Okay, remember that sugar cane is a C4 grass, toasted corn, C4 grass. Look in Coca-Cola, other than phosphoric acid, you mostly find corn products, as well as some sugarcane products. So we can actually, well, if we go to a different part of the grocery store, we can find some other things. So tomatoes, onions, bananas, mangoes are C3 foods. I just have an example here. This is teff, the Ethiopian flatbread, and some farmers harvesting teff and threshing it in the granaries. So human and other animal diet is related to what one ate. So we can apply this to the early hominins, early humanins, and ask the question, well, what did these guys eat? So one of these is a study we did on the so-called nutcracker man, and we looked at sort of a 1,000-kilometer gradient from Turkana and 3 other sites. And this is the famous nutcracker man who has this very large crest on its skull that's thought to be an attachment for the chewing muscles. And here's the diameter of the teeth of one of these guys compared to a human. Okay, so this is what your teeth look like. Look at those chompers. Really thick enamel. It's similar to gorillas in many ways, and the speculated diet since the discovery in 1957 by Mary Leakey of the first Synjanthropus was that probably tubers, nuts, or seeds, or something like that. And so we analyzed, there was no isotope analysis until about 2 years ago. And this is what was expected, and this is what we find. This guy was competing with zebras and warthogs for food. Okay, so this guy was going after a grass or sedge diet, but not what had been thought. And so this kind of makes a major rethinking, and just might point out the story of perseverance here. It took us about 20 years to get permission from when we first started working on this problem to actually be able to sample the fossils. It took about 10 years to convince the scientific community it worked, and then about another 10 years to convince the museums to sample their and these truly are very precious specimens. So rather than a diet, so those guys were in this kind of a habitat, probably eating. Baboons rarely do this, and this is why I took the picture. They're going after the sort of the corms at the base of the grass that are full of proteins for their diet resource. after we did the Paranthropus study, we were able to get permission to sample everything in the National Museums of Kenya. And this is a wonderful collaboration with the National Museums. And they said, well, okay, you can work on any broken tooth in the sample or in the museum. And so we came out with a series of all these papers last year, less than a year ago. And I'll have to admit that when we first started working on this, the amount of damage we did to teeth was significant, and we should not have been given permission to work on these teeth. But this is just one example. So this is a series of pictures before we sample and after we sample. So here's a broken tooth, and after I sample it, here's my sample in the bottom of the centrifuge tube. We're going to zoom in here. So this is the surface that I sampled. This is what it looked like before I sampled it, and this is what it looks like after I sampled it. So we knew we had succeeded when one of the curators said, now where did you take the sample after we'd done something? Then I knew we had them. They had to let us analyze things. So here's the story. 4 million years ago, our relatives were down in C3 land. This is almost a pure C3 diet, maybe experimenting a little bit with some of these C4 plants, but because a pure C3 land is definitely in here. We have other animals that are here. So they might have been experimenting a bit, but by 3.5 million years ago, an incredibly wide range of diets, and this is thought to be one species, but I'm wondering whether we're getting a differentiation into 2 diets, but we don't know yet. And then clearly 2 different, so this blue circles in the top are these Paranthropus guys, and this is early Homo. So by 2 million years ago, they had split their diets and were definitely not competing, maybe both arising out of this, maybe not. But in any case, their diets are totally separated. Here's C3 land, C4 land, which is basically maize, sugarcane, etc. So pure grasses. And we've got a very nice isotope distinction between them. If they were competing for space, they certainly were not competing for foods. So this is now our isotope story. And we're at the point now where we've now put down some of the rules that we can begin to speculate on what these diet differences were. But so far ourselves have kind of decided not to speculate on exact diets. So in summary, my odyssey in the last 40 years in working with paleoanthropologists has been trying to answer these questions about, these complicated questions. Well, how hot really was it? Well, it looks like these early humans, all the fossils that we have, all of them were living in really hot environments. Maybe they're living elsewhere. We don't have fossils that show that, though. We know they were capable of living in an environment like this, along rivers like this, in deserts, and we know now about some of their, and we know the sort of temperature ranges that they would be experiencing in open or closed or shaded environments, and now we can say something about their diet preferences. So I'll just close with one last summary. I can't kind of resist this. In the small little town of Archer's Post, I collect signs in Africa, folk art signs, and this is my favorite sign of all time, showing the evolution of humans at the Origins pub in the small town of Archer's Post, with dolphins swimming around, creatures coming out of the sea and evolving into early humans. And no doubt some of you will think that I've taken a wrong turn somewhere in my career, but I just don't believe road signs, all the road signs you have. So thank you for your attention, and I'm happy to answer questions.
shade and temperature, what they were eating. We want to know the species, Thure. Which species of plants were they eating?
Well, actually, some people have suggested that, other people, not me, that if we are very careful and can pick out the little plant material left behind in teeth, fossil phytoliths, we might be able to say something further on that. But I think that that's not what I'm doing, but I think it's something that in 10 years, the problem with most of these fossils is they've been so well cleaned that there's no phytoliths left. So that'd have to be done on new finds of fossils, but there's potential there. We may be able to do that. My colleague, Kate Freeman at Penn State, looks for what are called biomarkers. So these are molecules made specifically by individual plants. And so she's interested in working in the fossil soils to identify what sort of plant lineages might be there. And she's, I think, on the verge of some really exciting things in the next 5 to 10 years, but we don't have that yet. And we definitely don't have that preserved in fossils. Okay, well, so we've got a diet shift over about a 3 million year period. And during all of that time, we had these major rivers flowing through desert. But overall, the habitat was becoming more and more open. So it's the most closed early on, but it's still pretty open. And then as we continue all the way to the modern present, it gets more and more and more open. I guess the other thing that's fascinating to me in my kind of story that is a side story to this, but has been very important in our thinking, is this whole concept of the importance of C4 plants on the landscape. It's only been the last 7 or 8 million years. So the whole previous Earth history has had almost all of the photosynthesis is this what's called C3 photosynthesis. In the last 7 million years, the tropics have gone from maybe 1% C4 photosynthesis to 50% C4 photosynthesis. So these plants have really changed the whole ecology of the planet in a very short period of time.
On the dietary scatter plot summary, basically the time axis, sample points kind of grouped them so nicely, but what's the real uncertainty in the time axis and the dating itself?
Smaller than the size of the symbols. So in the Turkana Basin, so Frank could answer this better than I, but I think there's on the order of 60 different time horizons have been dated through the 3 or 4 million year interval that we're talking about.
So 3 million years.
So 3 million years, most of these would be plus or minus 50,000 years maybe. Yeah. So about the size of the symbol is the uncertainty of that, and we can just go back to that easily enough. Yeah. Yeah, so... Pardon?
How many points are pulled in here? 1,400 samples?
The 1,400 samples was the fossil soils, and that was from all over Africa for many, many different sites. So Turkana Basin, Olduvai Gorge, and many, many other places.
I think you've answered. I wanted to know about the parameter difference between Turkana and Olduvai.
One really important thing is Olduvai is and always has been, certainly in the record of time it's been there, has been much higher. So it's cooler by about close to 10 degrees Celsius, and it's simply because it's so much higher elevation. So, Olduvai is a much cooler environment, and I know my advisor at Berkeley, Dick Hay, was did the geology of Olduvai Gorge, and he went to Turkana and said, "I don't want to work here; it's too hot." Yeah.
So what role do the high temperatures of the Turkana Basin, what role are they thought to have played in human evolution?
Some people like Dave Carrier in the biology department here thinks that the high temperatures, that's a good way that they had to shed body heat, and so standing up and move, it gets you further away from the ground by standing up. And then he's also a proponent of that the early humans were runners, and this opening of the habitat, so the temperature would be going along with it, but having the more open habitat would be very good for people who run. And we know there are lots of running fans who I'm sure like that, think they're the top of the evolutionary pile. I think squash players are at the top. So. Yeah.
The hippos and the giraffes, were those samples taken from extant animals across the geographic range, or were they fossil?
So the calibration was based on modern animals, and then we've done the same thing in the fossil record. Okay. So what we try to do in the stuff that I've been doing is I try to have I and my students working in the modern environments so that we're well founded with what the problems are, as well as trying to work in the fossil record so that we can ground truth our hypotheses and methods.
Are those lineages ecologically consistent enough over time?
The hippos. So in the modern and the fossil, at least, we were both dealing with the same genus, what's thought to be the same genus. So there's some funny things that happen in the fossil record that we didn't get to talk about. But if we go back 2 million years ago, and if you think about African landscape today, you think, oh, wow, look at all of these antelopes all over the, and their relatives all over the environment, what we call the bovids. If you go back 2 million years ago, you find out, well, there was at least 3 different elephant creatures living on the landscape with, big, long, proboscided noses. And 2 of those were grazers and 1 wasn't. There were 3 pigs. Now there's only 1. You really find 1 pig on the landscape. There was 3 for a while. There were 2 coexisting horses, a 2 and a 3 and a 1-toed horse. And there are lots of other things that were grazers that are gone. And something has happened in the last 1 million years, and I wish I could tell you more. I think it's going to be a fun story to figure out. But almost all of the non-bovid grazers have been pushed off the landscape, in spite of there being more grass. We think there's more grass than there's ever been, but these other animals that were competing with the bovids have lost out for some reason. Yeah.
I was wondering if you looked at seasonality, like in the diet or in the temperature, or if you analyze one side.
Seasonality is something everybody wants, and we keep not getting it. And one of the things that we have thought, we'll get a tooth that grows continually. One of the problems is that the teeth, when they grow, they're not like a tape recorder and nicely record time. I know we don't have tape recorders anymore, but this is like a tape recorder where it's been recorded over the top of, and that's the way teeth form. And so they actually form over a fairly lengthy period of time, and it really messes up our signal. So we've kind of worked on some mathematical approaches to unscrambling this, and it really isn't quite satisfactory. I need more money and time to figure it out.
So you analyze one sample?
We take one sample like of a tooth, like of a horse tooth, and we analyze it sequentially. The problem is it's not a real big seasonal signal there today. There's a big temperature signal, but we need a big isotope. We needed some big isotope amplitude. And so we've done, it shows up nicely in Mongolia, but it doesn't show up nicely in East Africa. Yeah.
One of your charts sticks in my mind, one of the earliest ones, where you showed the C3 diet versus the C4 diet. And the C4 diet had things like sugar, and corn syrup, and all that kind of thing, basically. If we evolve with the C3-based diet, do you find any correlation to whether it's good or bad to have the C4-based diet?
Well, probably. I mean, we know that there's a lot of butchery going on from 2 million years ago to present. So our ancestors were eating meat, and that component could be coming actually from eating meat. So as far as we're concerned in the isotope lab, Tyson's chicken looks like a corn cob. For us, it's corn. And McDonald's hamburgers throughout most of North America, and this is the work of Leslie Chesson, who is a graduate student in the biology department, they look like they're 60 to 75% corn. So you can get this C4 component through meat if you're a omnivore or a carnivore. I don't know if that answers your question, but Denise Deering has said we should write it as the C4 diet book, and so we could become rich and famous and fund our research. She said this 12 years ago, and we haven't.
Still working on it.
Yeah.
Getting the recipes together.
So, but we've evolved over the last 4 million years, and our diets have included the human diet is on the order of 50% C4 based resources for at least 2 million years. So we've had plenty of time to evolve with it. But my gut feeling is you shouldn't only eat corn chips and nachos. The Super Bowl diet is probably not the best.
Would you be willing to put the aridity slide back?
Okay.
No specific question.
Oh, okay. Right. So what our idea there is that different animals get water sources in different ways. You can get it by drinking. You can get it by the water that's in your food. And Jim Elringer and I had a postdoc who actually used some of the findings that we worked on earlier to try to look at actually diabetes and how diabetes would change your isotope composition, and that actually we found some really interesting things there. So there's often unexpected sidebars. Now, I wouldn't say if we got an anomalous delta O18 value in the fossil record that I'm going to say, well, that guy had diabetes. I'm not going to go there. I'm not going to go there yet, but it did start with some laboratory animals that we had that we didn't know had diabetes that did have diabetes.
Well, thank you. That's good.
Mike,